Hello everyone. So today I'm going to talk about biochemistry subject, which is very important at GPAT point of view also, and which is very important subject of pharmacy. So in which I'm going to talk very short, that is related to carbohydrate and protein tests. Okay. So we will study what all other are carbohydrate identification test and protein identification test. So let's start with that. So yes, here you can see the carbohydrate and protein test. I have put the image of test tubes. So you will get how to perform the carbohydrate and protein test. Okay. So let's start with the studies. Reaction. And so I have put one column here which is very important as point of view of exams. So let's start with the reaction and the specific group of amino acid which is identified with the help of this particular reaction. So let me take a pen so you will get. And the first is biurate reaction. Biurate reaction is for to identify the two peptide linkage in the solution. Then inhydrin reaction followed by that menhydrin reaction mostly for the alpha amino acids. Then xanthoprotic reaction, that is for the benzene ring of aromatic amino acid, means mostly aromatic group identification, that is the amino acid like phenylalanine, tyrosine, tryptophan. So for the identification of those type of amino acid, we can use the xanthoprotic reaction. Then next is Millen's reaction. Millen's reaction for the phenolic group identification. The phenolic group is present in tyrosine. Then Hopkins-Cole reaction mostly for indole ring, that is a tryptophan. Then Sagakuchi reaction, Goniduno group, that is arginine. Then nitroprusside reaction, sulfahydryl groups, identification of SH group containing amino acids. Then sulfur test mostly for again the sulfahydryl group identification, but mostly specifically for the cysteine. Then Police test, that is for identification of imidazole ring, which is present in the histidine. And then the last one, Pauline Koik-Altu test, that is for the phenolic group identification, that is a phenolic group containing tyrosine. So here the most important reaction you have to remember, like all reactions are very important, but if you don't want to get confused, then Millen's reaction, that is phenolic group again, and Pauline's Koil-Catu reaction for the phenolic group again. So these two you have to remember because it will get confused for the identification of the phenolic group. And xanthoprotic, mostly aromatic containing amino acids can be identified with the help of xanthoprotic reaction. Okay. And then next is again carbohydrate test. Here you can see the Mollis test or also Barpo test. It is used for the identification of reducing sugars. So you know the example of reducing sugar, non-reducing sugars, so that you have to study. Then the iodine test mostly for the starches, starch identification. Then Benedict reagent test, that is for the, again reducing sugars. Then Selvinov test, ketohexose for the use of ketohexase. Folger test, that is ketohexose, rapid furfural test for ketohexose, Osazone test for the sugars only, general Osazone test mainly for the carbohydrates, means general identification of carbohydrate, and sucrose hydrolysis test, that is for the sucrose, and the name itself indicate that is for the sucrose. So here the important test is the Mollis test, that is for the reducing sugar, which is you have to remember. Okay. And the Benedict also for reducing sugar, and mainly you will perform these two tests in lab only, that is Mollis test and Benedict test for identification of reducing sugar. And the third and most important Osazone test, that is a preliminary test which is for the identification of the carbohydrate and protein, whether it is a carbohydrate and protein. So you can go for the Osazone test. Then Running Pharma by Priya, here my channel, and if you like my content, please give like, share and subscribe to my channel. My channel give you update about the pharma educations like all D Pharm, B Pharm, M Pharm and Pharm D. I'm going to make content for all pharmacy education curriculum. Then I will give you updates about the pharmacy career, what are the opportunities after B Pharma, after M Pharma and D Pharma. What are the pharma jobs available in Maharashtra and outside India as well. And yes, so that is my channel, and thank you very much for watching. If you like my content, please give like and share and subscribe to my channel. Thank you very much.